Hey team, Dr. Jack Audie here, and in this video I'm going to be taking you through this question. Why do we age? Now, whenever we talk about why in biology, there's always two answers. There's sort of the proximal why, the immediate why, which is molecular. There are molecular things going on and cellular things going on that lead to the aging process. Then there's the bigger picture why, and that is always referring to evolution. Why did we evolve to age, right? And the reason why we need to ask this question, I covered this a bit in the previous video, is that your skin, the surface of your skin is replaced weekly, and the deeper tissues are probably replaced every 10 to 15 years. And so your skin as an organ is 15 years old, max, which means your skin is the same age as Donald Trump's skin. So why does Donald Trump's skin look like that? And why does your skin look great and normal and smooth like mine, right? So it's clear that there's something else going on to what our intuition is, which is our intuition, everything breaks down over time. A car breaks down over time. But imagine a car in which you replace the parts continuously. It wouldn't break down over time because you've changed everything about it every 10 to 15 years, so it shouldn't break down over time. A car wouldn't. So why do we? And the hint there is I talked about evolution. Perhaps it is an intended process when it comes to evolution. So let's jump into it. Maybe you've heard of these before. These are quite popular. This is about the molecular why. I'll go into the evolution why at the end of the video, but molecularly, the process of aging are fascinating, and it does point to it being an avoidable process. We don't biologically have to age, which points to it having a purpose, evolutionarily speaking. So telomeres are these junk DNA at the end of chromosomes. Junk in that they have a purpose, they're made intentionally, they have a function, but they don't do what the rest of the DNA does, which is sort of program, have the blueprint for the body, for our body and of all biological functions, regulate DNA turning off and on, and creating recipes in the form of mRNA for proteins, right? So that's what the rest of our DNA does. So our telomeres don't do that, and they're in fact, evidence to their junk-like nature is that they're just a repeat of 6 nucleotides over and over and over again, right? So these nucleotides just repeat. And I'm going to go into why they repeat over and over again. But that gives you an idea that it's not really coding for anything. It doesn't contain instructions. It's just there as a protective cap. And that's what telomeres do. They're a protective cap to the ends of your chromosomes, to the end of your DNA strands. And we can see them here. They're fluorescent at the end of these little wiggly-like chromosomes there. And you can see these beautiful little fluorescent labels on our telomeres. They've just labeled that repeat there. So if you label that repeat, you can visualize the telomeres at the end of your chromosomes. So telomeres, what do they do? What do we mean by protection? To understand that, we need to understand a little bit about DNA and a little bit about DNA replication. So we all know that helical structure is like a twisted ladder, but ladders, we kind of think that the arms of the ladders go in the same direction. That's not true for DNA. DNA is anti-parallel. So there's sort of a chemical direction to DNA, and they run in opposite directions on the strand. So one strand goes this way and the other strand goes this way. So they're anti-parallel. Now, what happens during DNA replication? We unzip the DNA, then we bring in matching complementary nucleotides, so A to T, C to G, A to T, C to G. And if you unzip and you add the complementary nucleotides on, you end up with 2 separate strands of DNA that have the exact same code, right? So that's how we replicate our DNA. And we do that when cells divide. We need 2 copies. We need an extra 2 copies for when a cell divides. Cells already have 2 copies, so they need to make an extra 2 copies to have 4 copies, and then they divide, and now both cells will have 2 copies of your genome, of your all your 23 chromosomes. So you have 46 chromosomes in each cell. So when that happens, we need to replicate the DNA. So helicase unzips it. I always remember this by helicase sounds a bit like helicopter, and I imagine a little helicopter's rotor blades chopping the bonds between the nucleotides to unzip the DNA. So we're going to unzipping. Now, because of the anti-parallel nature of it, there is an enzyme called DNA polymerase that facilitates the new complementary nucleotides coming in and adding on to that unzipped DNA strand. Now, because of the anti-parallel nature of it, we've got a bit of a problem because DNA polymerase can only add to one end of the DNA, right? So chemically, DNA has a direction, and so DNA polymerase chemically has a direction. It can only work in one direction. So on one strand, it's nice and easy, and it can just, it's called the leading strand, and the DNA polymerase can just add nucleotides towards the unzipping. So as the DNA unzips, the DNA polymerase comes along in here and generates a whole new strand of DNA like that. And it's so easy because it's heading towards the unzip. But the DNA polymerase heading away from the unzip has a problem. So it duplicates this bit of DNA here. So now we've got a bit of DNA, but now a new bit of DNA is unzipped. So what the DNA polymerase has to do is jump over and head back towards where it was just working. So it has to work in chunks. And we call these chunks Okazaki fragments. So Okazaki fragments are these little bits of DNA that bind to the DNA and assist DNA polymerase mounting, and then it can go on extending the DNA polymerase. So it looks a bit like this. DNA, we get a little fragment there, an Okazaki fragment there joining, and then on the leading strand, the DNA polymerase can just keep heading towards the zip. It's super easy, right? It can just keep heading towards the zip. On the lagging strand, the DNA polymerase must mount it's gone. A primed piece of DNA with the Okazaki fragment, and then it can lead off, and then once it's done that small chunk, it must jump back and continue back towards where it was just working. So you can see here we've got a chunk, an extension, and then the DNA polymerase had to hop over and head back towards where it was just working, and it has to keep doing this over and over again. It has to go in circles. So now we're heading towards the end of the DNA. What happens once the helicase has split the entire piece of DNA? Let's have a look at what happens at that tail. What happens at that last little bit of the DNA strand? So here we've got it here. It's been completely unzipped by helicase. So this is the end of the chromosome. This is the last little bit of the DNA strand, and it just needs to be replicated and you're done. So on the leading strand, it just goes and flies off the end and it's done, right? So it can just beep, 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 and it's done. So that strand is fine. The lagging strand, DNA polymerase is heading in the other direction. So it heads along, boom, that animation didn't work out. Don't tell it with James Cameron, but yeah. Anyway, so the DNA polymerase heads along. Now it's hit where it was before. So now it needs to make that jump. It needs to go jump and then head back towards it. So it goes to make the jump, and it misses the DNA, right? It needs a certain length of DNA to jump onto. It can't just do a tiny little fragment. It needs that Okazaki fragment to bond, and that Okazaki fragment has a set length of nucleotides. So there's nothing for it to bind to, and so it doesn't replicate that last bit of DNA, so that disappears, right? That gets chopped off, and so now one of the versions of the DNA is a little bit shorter. So every time your cells divide, the DNA shrinks a little bit. Now, if we didn't have telomeres, we would have a functional gene here that wants to do something. It wants to create a protein. Maybe it regulates another gene. Maybe it's for a microRNA. It is a gene that does something, and now you've lost a chunk of it and it won't work, right? Maybe that was an essential domain of the protein. Maybe that was essential to the microRNA regulating other genes and so on and so forth. So you have now lost a functional gene, and that's going to have ramifications on their cells. What this means is that there's a limited number of cell divisions that your cells can go through, which is really, really interesting. And so your telomeres are a length of junk DNA there that can be eroded throughout your life without affecting function. They act like a clock. But what happens once they erode all away? Now you're going to get dysfunction. And remember, the definition of biological aging is a loss of function as we age. And we can see this happening molecularly right now. The DNA is losing its function because it's chopped off so much that it's gone into the coding regions and the functional regions of the DNA. So the repeated numbers decline as we age. So it's a great way to molecularly age your cells. You started with 2,500 repeats of this TTAGGGG that makes up your telomere. So as that repeat number declines, it tells you your biological age. But here's a question: why are we born super old, right? So your gametes, if you're a male, are constantly being produced. Your sperm die all the time, so you have to constantly produce new sperm, which means that that sperm is being created from a cell that's undergone loads of divisions, right? So surely then my sperm have short telomeres. I don't know if I should talk about one's sperm. If one is 40 years old, one would expect that their telomeres are short on their sperm, because the cells that produce sperm, that generate the sperm, will have had to divide and divide and divide, and as they divide, the telomeres shrink. The answer is there's an enzyme that creates more telomeres, and it's really, really clever. And now you kind of see can see why it has this repeated structure. So it's a protein that actually has a bit of RNA in it, and this RNA acts as a template and allows it to bind. And so we can see it's bound here. The RNA is complementary to the nucleotide sequence of the telomeres, and it's bound to it, C to G and A to T. And now this protein enzyme facilitates a whole bunch of new nucleotides coming in, A, G, G, G. And now you can see the enzyme can just slide along, and those 3 C's can now bind to these 3 G's. And then we can just keep extending the telomere, right? So it's a way to generate that telomere. It contains a template. It's a bit like a stamp, really. It's kind of just stamping more and more DNA in there, right? So you can see how by having this guide RNA start with 3 C's and end with 3 C's, it can just grab onto a piece of DNA, extend it, grab onto a piece of DNA, extend it, grab onto a piece of DNA, and extend it. So we have the ability to extend our telomeres. And this is where it gets interesting. So why not telomerase, which is the name of the enzyme? It's a great name. A's means enzymes, telomeres, A's, telomerase. Why not telomerase your whole body, right? Why don't we just extend the telomeres in every cell in our body every time we divide, right? This strongly points to it is an adaptive, evolved process, because we could not age our cells, but we do age our cells. Very interesting. So now we need to talk about the evolution, the big picture why. Why do we age? Because it seems to me that it's very intentional. It's an adaptive, it's a fit and adaptive evolutionary trait. It's advantageous to age. How could that be? So what I want you to do is I want you to, as just an example, this is just proof of principle example. Let's imagine 2 lion prides. Now each lion pride has one male, dominant male, that sleeps with all the females. Now those females create females and males, and when those males are old enough, they're actually kicked out of the pride. They go and grow up by themselves, and then they come back a little bit like the Lion King, and they take over the head of the pride in a battle, and then they become the king of the pride, right? So let's imagine a lion pride that doesn't age, right? The head of that pride, the king of the pride, the dominant male, won't age. So it gets first access to food, its building experience of its fighting techniques, and fending off challenges, and it's not aging. It is never going to be overthrown, or at least the rate of overthrow is going to be incredibly slow. You've got a giant, well-fed, well-trained male trying to be overthrown by a juvenile male that's eaten less, had to survive by itself, didn't have a harem of females collecting food for it, and has way less experience in fighting. So it's essentially the adults, the dominant male is going to outcompete its children, and so the DNA is not going to change from generation to generation. There is no there's no generations going on, and there's no genetic changing going on through the sexual reproduction process. Because they're not aging, they will outcompete their children, and we will just end up with a very stagnant gene pool, as we're not getting generation after generation after generation. An irregular line pride that ages, the dominant male will have more experience, will be well fed, but it will start to age. It will start to lose functionality. And so now it can't outcompete its children, and it will be replaced. It will die and be replaced, and the next generation will come through. So they're going to go through generation after generation after generation. Now imagine the gazelles that they're chasing, all age in both situations. The gazelles against these lions are going to evolve to be faster and smarter, but these lions aren't going to evolve because they aren't having those generation after generation, right? And so this lion will end up being very slow and die, and that whole species will die out because it's not got enough of a tick over after generation after generation. Right, so here we can see that evolution seems to be an adaptive process to prevent parents from outcompeting their children. You need generation after generation for survival of the fittest to work on, and also this process creates genetic variation. And if you don't have that, you're going to have a narrow gene pool. You're going to outcompete your children. They're going to die off, and so you're not going to be able to evolve essentially because you're not having generation after generation. And so now we can see why, I mentioned this in a previous video, parrots that live very fast lives, they beat their wings, they have a high heart rate, they're very active, they can live to 70 years old. Lions sleep 23 hours a day, their heart rate's really slow, and they live to 10 or 12 years. And so you can see that it's not about how fast you live. It's not about being exposed to the environment. It's not about being small things live for a short amount of time and large things live for about a large amount of time. It's about what does the evolutionary niche that you fit require? If you look after your young, maybe you have to live longer, right? So you will age slower, right? So it's an adaptive process that can be designed to fit the niche and biological lifestyle that you live. So the mayfly that essentially just comes out and breeds, and creates eggs and dies after 24 hours, it ages incredibly quickly. It doesn't look after its young. It doesn't have to learn how to hunt or any of that kind of stuff. So it ages incredibly quickly and it dies after 24 hours, right? Humans, we look after our young. We're completely dependent for like decades. Like, I bet many of you are still living with your parents, right? So, maybe a decade and a half, we're dependent on our parents. And so we have to live longer, right? And so we've evolved to age slower. We have longer telomeres, for example. Our cell division cycles are slower. Our telomerase activity might be higher, this kind of thing. It's an intentional adaptive process. So what else is going on? Because there is a little bit more to aging than telomeres. So there's senescence. Senescence is cells dropping out of the cell cycle after a certain number of replications. So basically we found if you take cells out of the body and you grow them in a nice media so they've got all the fuel that they could possibly want, after about 30 to 40 cell divisions, they stop dividing, right? And so this is a process called senescence. And so cells that stop dividing build up in your body, and so you lose your regenerative capacity as you age, right? So a very simple example is wound healing. If you have a wound when you're very young, your cells haven't senesced, so the regenerative properties of your skin is huge. A huge number of cells have the ability to divide and heal that wound. If you injure yourself when you're old, your intrinsic proliferative properties of the tissue are massively down, hugely down. And so it's a slower process, the healing process. It's disorganized. It requires the recruitment of cells. Fibroblasts come in and we end up with a scar, right? So senescent cells dropping out of the cell cycle, that they build up in our body. But here's a question. What tells the cell to drop out of it? One of the major signals is the telomere length. So here is the telomere in red. We can see it shrinks after generation. And it's after 40 generations, the telomere has shrunk so much that it signals to the cell to undergo senescence. You cannot survive another division without damaging your DNA, so stop dividing and just sit there and function, right? So that is senescence, and it's still tied to telomeres. But there are certain processes that are a lot more like a rusting car and a little bit less like a controlled process. And one example is reactive oxygen species. So by breathing and using oxygen in our metabolism, we produce very reactive chemicals. And very reactive chemicals react with other chemicals really readily. That seems obvious. Those other chemicals are trying to do something in our body. Typically, they might be a protein, they might be a lipid. They're trying to do something. So when a reactive oxygen species reacts with it, it changes its chemical composition. Its chemical composition dictates its structure dictates its function. So reactive oxygen species, by reacting with molecules, block the function of that molecule and damage it. Now, you might just think, well, couldn't you just replace that molecule? Well, what if that molecule was DNA? Or what if so many molecules got damaged that the cell died? Now, that's normally not a problem because you have proliferative cells that can heal. But what about in tissues that replace incredibly slowly or don't replace or barely replace at all? Some examples of this are heart muscles. These almost never replace themselves. There's no intrinsic proliferative properties inside the heart or the brain, or inside also your hearing cells inside your ears. So if you get a reactive oxygen species burst in any of these places, it could cause damage to the cells that can't be replaced. You cannot replace a hair cell. It's very difficult to replace a neuron. It's very difficult to replace a heart muscle cell. So we can, in that respect, get that sort of more intuitive form of aging where we get a slow buildup of damage throughout our life. Another kind of thing that's a little bit more intuitive is the buildup of persistent pathological material. There are certain biological processes in your body that create something that we can't get rid of ever. And so it builds up in our body and causes dysfunction to things around it. Two classic examples are cholesterol crystal and lipid deposits inside our blood vessels. We're very bad at getting rid of that, so it slowly builds up in your life. It actually starts building up very young. Autopsies during the Korean War showed that 18-year-olds had these atherosclerotic plaques in their blood, and so they're already undergoing the aging process as they build up plaques in their blood. And eventually it can cause a clot, which leads to things like heart attack or stroke, and also dysfunction, right? You've got lower blood flow going on. Another example is Alzheimer's disease and other protein aggregate diseases. And this is where proteins aggregate to the point that we can't break them down again. And so we build up these protein aggregates. Now, this is just a theory and it's so complicated, but in Alzheimer's disease, we do get this buildup of protein aggregates in our brain, and it does cause some problems. Whether it is the ultimate cause of Alzheimer's disease, we're still trying to figure it out. But here is there's a Golgi stain of a brain, and we can see here's some neurons coming down here, neuron. And here we can see this giant protein aggregate building up in the brain there. And this is made of amyloid, and amyloid is this protein that aggregates during Alzheimer's disease. So because it's a process that generates a persistent material that we cannot get rid of, it ultimately slowly builds up dysfunction throughout our life, and that is part of the aging process too. Of course, this all begs the question, why didn't we evolve to be able to get rid of these building up compounds? Why can't we get rid of the cholesterol? Why can't we get rid of the amyloid that's building up in our brain? And the answer is we're supposed to age anyway. We've already raised our kids to adulthood. We're undergoing the aging process. It's not worth the energy to get rid of it because we've been sort of programmed to die around that point anyway. So it doesn't influence the survival of our children if I die of a heart attack at 75 or 80 years old, right? So it doesn't affect the success of my genes going on into the next generation. So aging, here's a man, he's got loss of function, which is the definition of biological aging because he's stooped and he's got a walker. And there's a number of things that can lead to this aging process. We discussed telomere shortening. There is genetics, and so certain people have a genetic makeup that resists the aging process. Other people have genes that have been turned off and turned on to orchestrate the aging process, and different people have different genetics that orchestrate the aging process at different rates, right? There's reactive oxygen species that is a natural process of breathing oxygen and using it in our metabolism. There's cell senescence, and that's all tied to telomeres. And then there's a process called inflammation. Now, I love inflammation, and what we've found recently is that inflammation actually links to all these processes. If you have inflammation, it accelerates telomere shortening. Inflammation turns on some of the genes associated with aging. Inflammation definitely releases lots of reactive oxygen species, and inflammatory cytokines actually signal for cells to undergo senescence, right? So inflammation is highly linked to all of the aging process. And what we've also found is that as we age, our basal level of inflammation increases, right? And so we call this process inflammaging, right? So maybe the raising level of inflammation as we age is being a master controller for the aging process. Further evidence for this is when we look at things that seem to accelerate the aging process, like a bad diet, infection, smoking, alcoholism, a lack of exercise, and obesity all seem to accelerate the aging process. And many of these things enter in the molecular process of aging through inflammation. We know that poor diets are inflammatory. We know that infections are inflammatory. We know that smoking causes inflammation. We know that alcoholism causes inflammation, particularly of the liver. We know that obesity causes inflammation and causes chronic inflammation. And we know that exercise can lower inflammation. And so a lot of these things seem to enter the aging process through inflammation. There is also other things going on. Smoking causes reactive oxygen species, for example, and so do infections. And so there are a number of environmental factors that can influence biological factors which can change the rate at which we age. And it might just be, this is me, and I believe inflammation is the center of the universe. So I think inflammation is one of the master switches of the aging process. And that's just me because I'm an inflammation researcher. If you ask a different researcher who researches, say, free radicals, they'll say it's reactive oxygen species. But yeah, those are key molecular processes involved in the aging process, and that's how the environment interacts with it.